When a cell divides, its genetic code is exposed. A single ultraviolet ray, a chemical toxin, or a microscopic error during replication ruptures the DNA sequence. In that fraction of a second, the lineage enters a biological crisis. For decades, basic biology courses have taught that the protein p53 responds to this crisis by acting as a cellular brake. That analogy is deeply inadequate. Cells do not simply drive and stop. This diagram maps the actual architecture. p53 operates as a highly active transcription factor, a specialized protein that binds to DNA to control which genes are turned on or off. It sits as a central integration node, constantly receiving incoming data streams about metabolic strain, nutrient availability, and DNA integrity. The protein does not carry a single fixed command. It computes these variables. Based on the intensity and duration of the stress, p53 weights the outputs and drives the cell down one of three severe paths: temporary repair, permanent arrest, or immediate death. p53 is a biological decision architecture. It governs cellular fate through strict calculation and graded outputs, evaluating risk against the biochemical cost of survival. That calculation contains a severe evolutionary tension. The exact mechanisms p53 executes to suppress malignant tumors early in life are the primary drivers of physical tissue degradation as we age. Furthermore, when the genetic code for p53 mutates, the node does not merely break. Specific corrupted forms of this protein actively rewire the cell's logic, becoming the greatest survival asset a tumor possesses. Deciphering p53 requires looking past the desire to simply halt a tumor. It demands an understanding of the profound biological cost of survival and the strict, silent intelligence embedded in the regulatory state of our cells. Living systems rely on conditional logic. p53 constantly reads the cellular environment, translating the severity of stress into a proportionate genetic response. In the presence of minor, correctable DNA damage, p53 activates a specific set of target genes. These genes halt the cell division cycle, granting the biological machinery a narrow window of time to physically reconnect severed DNA strands. This graph illustrates what happens when the damage surpasses the cell's repair utility. The biochemical pressure climbs steadily along the x-axis. The cell tolerates the accumulating stress until it hits a rigid mathematical limit, a point of no return. At that exact peak, p53 shifts the balance of the BCL2 protein family. These are specialized regulators that determine the stability of the cell's energy centers, the mitochondria. By tipping this balance, p53 triggers mitochondrial outer membrane permeabilization. The mitochondria physically tear open, releasing lethal proteins into the cell. This is the definitive, irreversible execution order for apoptosis. This violent dismantling is a highly coordinated sequence. Every step is executed with precision, ensuring the dying cell is absorbed without triggering a chaotic immune response. Apoptosis is a principled containment strategy. The individual cell is sacrificed to protect the structural integrity and genetic fidelity of the wider organism. Death is not p53's only method of containment. When a cell faces persistent stress that falls short of the lethal threshold, the system initiates an alternative protective measure: cellular senescence. Senescence is a state of durable, permanent arrest. The damaged cell remains metabolically active. It consumes energy and produces proteins, but it loses the mechanical ability to divide ever again. Evolutionarily, this logic is ruthlessly effective. By permanently locking down cells that carry dangerous mutations, the organism effectively suppresses tumor growth long enough to survive its reproductive window. But biological safeguards exact a toll. As tissues undergo decades of routine turnover, these dark, senescent cells accumulate, permanently lodging themselves into the local architecture. This cross-section of epithelial tissue demonstrates the immediate danger. The senescent cells, marked in orange, are not dormant bystanders. They actively secrete a continuous stream of inflammatory factors that actively degrade the healthy cells surrounding them. This localized inflammation eventually dictates the aging phenotype. The toxic environment exhausts the functional stem cell pool. The tissue becomes rigid, slow to heal, and physically scarred. This forces a clinical reality check. Attempting to synthetically dial up p53 vigilance to perfectly suppress malignancy would inevitably accelerate this widespread physical decay. The strict cellular governance that protects us from cancer in our 20s slowly bankrupts our regenerative capacity. Aging is the visible residue of our own survival mechanisms. Malignancy is routinely misunderstood as a disease of absence, a scenario where tumor suppressors simply break, leaving the cell without hardware to stop division. While simple genetic deletions occur, cancer relies on a much darker adaptation, known as a gain-of-function mutation. In a gain-of-function mutation, the genetic code for p53 is altered, causing the resulting protein to fold incorrectly. Instead of being cleared away, this highly stabilized, corrupted protein accumulates in massive, suffocating densities inside the nucleus. Looking back at our network diagram, the consequence is immediate. The toxic node severs the standard biological logic. The commands for permanent arrest and cell death are physically disconnected. But the protein is still an active transcription factor. It begins rewiring the network, generating aggressive new pathways that stimulate metabolic adaptation, tissue invasion, and therapy resistance. The tumor cell weaponizes this corrupted node. When the cell encounters severe stress, like the DNA-shattering impact of chemotherapy, the mutant p53 intercepts those alarm signals and converts them into instructions for malignant endurance. A tumor with a simple loss-of-function mutation is dangerously blind to damage. A tumor with a gain-of-function mutation is actively instructed on how to survive it. The disease is not a mere breakdown of cellular structure. It is an organized exploitation of the rules. The very molecule that evolved to guard the tissue is forced to organize its destruction. TP53 is the most frequently altered gene across all human cancers. The obvious clinical ambition is to synthetically restore its function. Yet, restoring a regulatory network is entirely different from fixing a mechanical switch. The pharmacological roadblock: we fight absent proteins, paralyzed proteins, and mutants resisting treatment. Over time, this expands into a subclonal phylogenetic tree. Singular therapy creates an evolutionary bottleneck. The main tumor dies, but subclones harboring escape routes survive, multiply, and repopulate the mass. Modern oncology treats p53 restoration not as a standalone cure, but as a strategic maneuver. Reawakening even a fraction of the cell's internal discipline strips away the tumor's adaptability, exposing fragilities that combination therapies can exploit. Beyond the clinic, the pharmacology points back to the central biological reality governing our tissues. Complex, multicellular life does not sustain itself through unchecked vitality or endless replication. It endures through measured refusal. p53 proves that biological continuation requires an internal chemical intelligence. A cell must possess the capacity to interpret its own damage, calculate the risk, and yield its own future to ensure the survival of the whole.